This conference will now be recorded.
Okay, welcome everybody. My name is Kristen Musselman. I'm a scientist and assistant professor at the Toronto Rehab Institute in the University of Toronto, and it is my pleasure to moderate today's webinar. Before I introduce today's speaker, I have a few housekeeping details to review with you. First, please mute your phone line or your mic, computer mic, throughout the duration of the presentation. Second, we will have a question and answer period at the end of the presentation. Feel free to type in any questions you may have as Dr. Clanton goes through his talk. I will keep track of those questions and bring them up in the Q&A period. So it's my pleasure to introduce Dr. Samuel Clanton. He is an assistant professor in the Department of Physical Medicine and Rehabilitation at Virginia Commonwealth University and a physician at Sheltering Arms Physical Rehabilitation Hospital in Richmond, Virginia. Dr. Clanton has a PhD from Carnegie Mellon University in robotics, jointly with the CMU Center for the Neural Basis of Cognition, and a medical degree from the University of Pittsburgh. He later completed a medical residency at the Rehabilitation Institute of Chicago, followed by a fellowship in traumatic brain injury/polytrauma at the Hunter Holmes McGuire Richmond VA Medical Center and Virginia Commonwealth University. He's worked in brain-computer interface research first at the NASA AMS Research Center as a staff researcher earlier in his career, and later during his PhD research, in which he was able to show high-fidelity control of a 7-degree of freedom robotic arm using a primate brain-computer interface model. He now treats inpatient traumatic brain injury and other rehabilitation patients at Sheltering Arms, and is part of the Sheltering Arms Institute, a new collaboration between Virginia Commonwealth University Physical Medicine and Rehabilitation, and Sheltering Arms. And I want to say, give Dr. Clanton a big thank you on behalf of the SCI Special Interest Group of the ACRM for presenting with us today. Thank you.
All right, thanks a lot, Kristen. I just want to say thank you to Kristen and everyone here here to watch me speak today. In any case, I'm, this is an interesting talk to give to the spinal cord injury group, FES task force, because I'm a brain injury guy, first of all, not spinal cord, and then also I've never worked with FES specifically. However, I think this is a topic very relevant to a lot of different FES devices that can be used with brain-computer interfaces, and hopefully I'll shed some light on sort of the input mechanisms that can be used with FES setups for brain-controlled prosthetic or brain-controlled movement of the body. In case my background, Kristen mentioned, I really, clinically, I work as a PM&R physician, mostly in brain injury now, but I really kind of started out thinking I would be a researcher, kind of worked with brain-computer interfaces extensively, first for NASA with EEG, and then at CMU and Pitt on some sort of higher fidelity, really interesting brain-computer interface experiments, which I'm going to present as a part of this talk today. Specifically, what I'm going to talk about is really I'm going to give an intro for people less familiar with brain-computer interfaces. Probably a lot of people have some familiarity with it here. I'm going to talk about what they are, kind of how they work for specifically electrical, systems to record signals from the brain electrically to control external devices, and then kind of talk about their potential in rehabilitation for people less familiar. And then what I really want to get into is how, a lot of times I'll see in research, especially in the news and videos on the news, they'll talk about something being brain controlled, and it's all kind of equivalent. There are different recording mechanisms that are used with different devices, and a lot of times they look similar. People are controlling, for instance, in my research, a robot arm using very different recording interfaces and producing a robot arm that moves and produces grasping behaviors and really complex behaviors. But if you really look at it and think about what's going on and know the research, you know that what's actually being controlled by the brain-computer interfaces is very different. So I'm going to talk about how the different types of recording interfaces lead to different types of control signals that can really be used in different ways. I'll talk specifically about how these control mechanisms are used to be transformed into machine commands, and then really what our expectations should be when we're using these different devices. I think right now, especially seeing this clinically, people don't really understand that some of the using a simple setup for control with BCI kind of can lead to kind of more simple results, at least in the current state of technology. And then, so I just want to make sure the people have adjust their expectations of what different BCI recording mechanisms are able to do. And I'm going to use examples first from my own research in cortical microelectrode BCI recordings, and then some examples externally with EEG brain-computer interfaces that are very good, but just a different nature. So in any case, no disclosures. So the idea of brain-computer interface prosthetics. So again, I'm going to focus on electrical signal recording, primarily EEG and microelectrode, actual electrodes implanted in the brain. There's a variety of different sort of setups that have been used to work with BCI, things like fMRI, some optical things, some magnetic MEG, things like that, but I'm going to concentrate on electrical recording. It's the most common. And then also specifically with control of upper limb prosthetics, just because that's kind of my background and what I'm familiar with. Lower limb is very different and different. There's spellers, communication devices, things like that, a little beyond the scope of this talk. So essentially the idea here is that we capture electrical activity in the brain directly. Neurons communicate and control the body through electrical signals. We capture those electrical signals at their source. And then in the second step, we quantify those signals. So one way or another, we turn those into a sort of, you can think of it as a vector of commands. You could think about what, different degrees of freedom or different independent expressions of some wish or command from the brain. The third step is then decoding this command into a set of commands in the end effector space, the robotic space, the control degree of freedom space. So that input signal dimensionality or number of independent commands can be different than the set of commands within the robotic control space. So for instance, I've put a few examples of end effectors that can be controlled using a brain-computer interface. First is a robotic arm from my own research. Second is an FES, just an illustration of an FES setup used in a brain-computer interface. In my experiment, we used the control of the end effector control, so the position, location, and orientation of a robotic hand in space. And the second, in FES experiment, it's often muscle activity of individual muscles. So there's an input kind of dimensionality, input degrees of freedom, and then output degrees of freedom in whatever control mechanism you're using for your device. And the idea here overall is to restore function to people with ALS, spinal cord injury, for whatever neuromuscular or other conditions where people, for one reason or another, have an intact brain, cognition's intact, the desire to move their arm is there, but for one reason or another is disconnected from their arm itself. The idea is to replace that connection from brain to muscle or brain to peripheral nerve in order to restore function, or a brain to external robot to be able to interact with the environment in the same way people were before their injury or illness. All right, so just to start out, just to make the point directly, that different electrical BMI or BCI recording systems are very different. We kind of have two ends of the spectrum. One is EEG. Place electrodes on the head. Very simple. You can do this at home to some extent if you want. Used clinically commonly, non-invasive, etc., relatively cheap. On the other end of the spectrum, we have single unit recording. These are actual microelectrodes implanted into the brain. These electrodes record directly from often individual units within the cortex. So the difference is, besides their level of invasiveness, they are actually recording very different things. EEG is recording from roughly a 3 centimeter area in the gyri of the brain, kind of around the electrode. It really can only pick up kind of things that all the neurons are doing together, or the predominant signals from huge populations of neurons. At the other end of the spectrum, single unit recordings, a single unit action potential recording is often individual neurons or maybe a few neurons that they're closely linked and activating together. In between those things, we have some other recording mechanisms such as ECOG. ECOG is electrocorticography. That is basically placing kind of EEG-like electrodes on top of the dura or below the dura, on top of the brain, non-invasive to the brain, but still an invasive procedure, as well as local field potentials, which are using implanted microelectrodes, but picking up a larger population of neuronal activity. So you kind of have this trade-off between ease of use, cost, and ability to get at what individual neurons are saying. And so the difficulty is very different, but the signals you get out are very different. So I'm going to start by illustrating kind of the fundamentals of single unit recording and our experiments with this. So this is kind of taken from an older work just on how single unit recordings record individual neuronal signals. There's a very, very small microelectrode in the brain. You're looking at real-time voltage outputs at the electrode, and then if you have you look at the voltage deflections, and you could say, okay, this is an individual neuron spiking. You can then represent that spike as a discrete element in time. So you say, okay, at time, each time it spikes, you talk about that being a particular trigger or a tick mark in the C diagram up above. You then can overlay, say, a population of neurons, and that's illustrated in D. So we have many neurons firing at the same time, and sort of you looking at a time sequence, and that concentration of tick marks says that neurons fire more often. And for instance, if you have a stimulus being presented to the person or subject or animal, you can then record the neuronal response to that activity, or say you've given a movement command, or you have a timing sequence to elicit movement in a subject, you can look at that neuronal fire and response. You can then kind of sum these into a histogram and look at the event-related discharge of the population of neurons. Now, what we can do then is translate this activity of a single unit or single neuron or a population of neurons into a quantified signal of spikes per second, just a spike rate of an individual firing neuron. And that's used as the fundamental basis for many, at least that I'm familiar with, many of the inputs for brain-computer interface systems using microelectrode recording. Now, there's been progress over the past, at this point, 20 years in brain-computer interface prosthetics that is using these types of systems to control external devices. This started kind of around turn of the century, which is weird to say in the 21st century, but with basically an experimental setup of moving cursors in a virtual reality screen. So basically a 3D virtual reality environment. Monkeys were used to, first, they would actually be, the cursor would be controlled by their hand. They would have to reach one of 4 or 8 targets, and they would then record brain activity during that arm movement, and then they would start controlling the cursor not with the arm movement, but instead with the brain directly. And that's kind of how this started. And that's more like a target acquisition, just kind of cursor in virtual space experiment. Next, a robot was added to the loop. So there's a monkey still looking at a screen with cursors, trained in arm movement, but now he's controlling a robot arm, and the output from the robot arm provided the location of the cursor in space. So this was, one of the first experiments with brain-computer interface control of a robotic arm. And then later, we started getting into, and this is now in laboratory I've been associated with, getting into more arm behaviors. In 2008, Felice et al. Published a paper on a self-feeding experiment with the monkeys, first trained in arm control, kind of similar, but then no longer using cursors, just using the arm. He was able to feed himself marshmallows. And now with the 4th dimension of movement of closing the gripper using brain control, able to successfully perform this activity. And then later, in the later experiments, there was an increase of more sophisticated behaviors. And so I'm going to present an experiment that the beginnings of which are shown on the right, where we wanted, first of all, full control of an arm, not just 3D locations in space corresponding to cursor control, but now full control of rotation of the hand and reproducing the full dimensionality of the arm, of our own arms. We did control the robot in the space of the hand and its location and orientation in space, as well as gripper control, and then overall in 7 dimensions of control. We also wanted to not have arm movement be a part of this at all, to make it more applicable to people without the ability to move their own arms for any kind of cortical training, and kind of go through how we're able to do this experiment. All right, so how do we get commands in the space of the robot from, how did they get the monkey brain signals to control the cursor? So the way this started in the initial experiments using cursors is based on the observation that when monkeys move their own arm, certain neurons fire more than others in certain directions. So for instance, say we have a population of neurons that's only two neurons. We have neuron A and neuron B. A monkey is moving his joystick or moving his arm in general. Neuron A essentially fires its fastest when the monkey moves his arm forward, the least when he moves his arm backwards, and somewhere in between when he moves left and right. And then neuron B has a different direction that it prefers. So it fires maximally when the monkey moves his arm to the right, the least when he moves it to the left, and somewhere in between when he moves up and down. And you can see how we could then decode activities of these two neurons and translate that into two-dimensional movement control. Now for our experiment, this actually can then be extended into 3 dimensions. And the way that a full population was put together was thinking about this like a sort of a voting scheme. So individual neurons have preferred directions. They would then multiply, you can imagine as a vector, multiply that vector and scale that vector by the relative firing rate of an individual unit. And then they would essentially take that output from all the different units and sum them together. And essentially that output vector, the summation of all those individual units, became what was called the population vector. And that was kind of the voted direction by all the population of neurons. So this was extended to 3 dimensions. And what we wanted to do was actually extend this to n dimensions, as many dimensionality control, that being the full control of, in our case, linear motion, rotational motion, hand movement, etc., just kind of all tacked together into one model. As long as the axes of this control system are orthogonal, the scheme for doing this should be successful. So what we did is we made a consistent mathematical framework for producing something like the population vector. But what was happening, especially in higher dimensional space, is certain directions in that 6-7 dimensional space were more highly represented than others. And so using a mathematical technique called optimal linear estimation, we're able to essentially normalize that data and reduce that bias from different neurons in the population that preferred certain directions more than others. So essentially being able to sample evenly over the entire command space instead of the just preferred by chance sampling of a large population. I won't get into the specific mathematical details, but we're able to create a set of linear algorithms. They're able to do this without complex regression, simple linear transform that was able to output a set of n-dimensional predicted movement commands from a set of real-time input spike rates going on in 30 millisecond bins is what we ended up using, sort of in our case, based on a set of weights that would produce that output. All right. So in this experiment with a monkey, in the prior panel, we implanted Utah 96 electrode microelectrode arrays into the approximate, as close as we could get, motor cortices of monkeys, and we'll show how successful we were on that later. That's one of the arrays now on my fingertip. This is one of the intraoperative photos, and then we would get these deflections in voltage out of each electrode. And we're actually picking up, in many cases, more than one unit or more than one neuron or closely correlated neurons at each electrode. And we would actually manually separate them. They kind of come in differently to the electrode based on their location and distance from the electrode. And we would separate them using bins, and so we could actually record multiple units at each electrode. Then we'd count how many spikes per second were coming into each of those bins, count that as a spike rate, the deflections per second. And these are the spike rates we use for robot control. Here's our experimental setup. We had a monkey. His implants are not shown here. We had a robot, kind of a giant robot compared to him, a Barrett whole arm manipulator WAM arm mounted to his right. He would essentially be controlling this robot. Second robot was an industrial robot used just to present a target cylinder. at the start of each trial, and he would initiate trials by pushing a button in front of him, would move hit the arm he would then control into one of 6 starting locations, as simultaneously the targeting robot would move the cylinder to one of 6 orientations. And so the monkey would have to, in real time, control the robot to move to a destination, position, and orientation. And this was evenly distributed among the 6 dimensions of movement. At the end of each trial, he would also have to open the gripper and then close it around the cylinder to complete each trial. He'd receive a water or juice reward at the end of each successful trial. And we didn't want the monkey to ever move his arm himself. It wasn't trained for arm movement in an experiment similar to this. We wanted him just to be able to kind of do it without having to produce movement on his own, just to simulate kind of clinical conditions. So we actually just use observation of the robot to calibrate our neural decoding models. So here we're watching, this is not brain control. This is automatic control. He pushes the button, goes to the starting location. Now this is automatic control towards the target. So the robot's moving autonomously towards the target region at the correct orientation and location, grasping the cylinder, and we are recording from the monkey's brain each time that this is occurring. And this is the basis for the decoding algorithms, what we use to calibrate our neural decoding algorithms to later perform brain control. Now, at this point, considering this experiment was ultimately successful, it raises a few questions about what was going on here, because there are a lot of theories about things like motor neurons mirror neurons or mirroring systems in the motor cortex. It makes you wonder if this monkey saw this robot as an arm. Was this giant non-monkey robotic arm something that seemed like an arm to him and could be understood subconsciously as controlled, kind of directional control? Not sure, but one thing we'd also noticed is if we're in the room and we're listening to neural spiking going on in the background, which is commonly the case, we would come in between experiments to kind of fix things, do maintenance. There's all kinds of robotic issues and technical issues throughout. We would notice that his spike patterns would sometimes correlate with our movements too. So kind of depending on directions, kind of random or different tools we're using and things like that, we seem to see a correlation with different activities. And those were very different between different neurons, even neighboring neurons in different populations from different electrodes. So some really interesting stuff here. So in any case, at this point, we calibrated our neural decoding algorithm, and that raises another question: is how do we then get a monkey to do this? This is kind of a complex experiment for a monkey to be able to complete. 7-dimensional simultaneous brain control is not something we felt we could drop the monkey into immediately. Monkeys don't know what you're trying to do. They kind of don't care about what you're trying to do. So we needed a way to kind of introduce them gradually. And we did this via a type of shared control, that is sort of collaborative control between the machine and the human, or a monkey in this case. So we essentially created a virtual kind of perfect controller that at all times would be kind of pointing towards the target region. We would then kind of split control between the monkey and the virtual monkey or virtual controller. And we did this in 2 ways. And there's a reason for this. It's a little complicated, but there's a reason for this. The first way is we just added together the commands from the computer and the monkey. So even if the monkey didn't do anything, it would ultimately hit the target. The monkey really didn't have to participate, but he could start to see that he was able to influence the movement of the robot. And that was used, first of all, for the observation trials, used for calibration, and then also as sort of the initial trials with some brain control to help establish that link between his control of the robot, but deliver a consistent reward to the monkey. Monkey attention and participation was probably by far the hardest part of this whole experiment. They very short attention span, and we really had to maintain this kind of level of providing consistent reward, but also maintaining challenge so he would get better at this in order to ensure success in the end. The second way, which was kind of more important for kind of after the beginning stages of the experiment, is we would actually use a concept called virtual fixturing. And this really just eliminates error in a proportional ratio or kind of controllable proportion from the monkey's command. So we actually developed fixtures. Virtual fixtures are a concept from haptic research and as well as kind of robotic telesurgery to kind of bound error in human input within a control, kind of a movement control. But a lot of these algorithms were only in a couple, 2 or 3 dimensions. So what we had was essentially an experiment where the monkey's controlling a 7-dimensional, has a 7-dimensional control space. And so we figured out a way to kind of bound error within that high dimensional control space by projecting onto the manifold of allowable commands. Essentially, I'm showing a cylinder here, but it was all points around a cylinder. In reality, the successful command space was really anywhere around the cylinder that he was able to grasp to successfully complete a trial. So we would obtain via this algorithm a projection to this manifold in 7 dimensions. I'm showing a 3D representation in the upper right. And then we would adjust the proportion of allowable error away from that manifold. So the monkey would either, the green arrow up in the top right shows the monkey's raw input velocity. The red arrow shows the projection onto the manifold, and there's a yellow arrow, which actually I'm seeing maybe hard to see, which is 50% error allowance, essentially in an instantaneous command. And below I'm showing a trajectory basically from the blue line is towards the target region. That's a sphere again. The blue line is the raw data input by the monkey, and then with differing amounts of error allowance, it would basically be bounded. The overall control would be bounded closer and closer to the target. So by this way, we were really able to get the monkey to control the arm, but bound the error to kind of shape his behavior so it would end up at the target so he could receive a reward. And then we would decrease the amount of error reduction over time as he got better at the device. If we had a long period of no reward delivery, the monkey he just didn't really care what we were doing. You could tell it was very difficult. Every time there was a successful trial towards the end, when it became more difficult, he would take a break and then kind of get back to it. We were glad we introduced the button to show that he was attending to the task, but it made recording videos of the control very difficult. But in any case, we felt like this really allowed the experiment to, rationally, he was able to rationally kind of learn the experiment without really understanding what we were trying to do. Another element of this was we implemented a novel impedance control algorithm in the robot, so he was able to interact with the device. This is not full brain control yet. Some trials might be, actually, I'm not actually sure, but this is him interacting with the device. This is kind of an introductory. It's the first time we'd shown kind of object interaction, kind of force interactions, impedance interactions between the robot and objects in the environment. And he was able to control through this. Essentially, when there's a collision, it became not really force, but a control of how much effort was given kind of to go through the object, and he used that to tilt the wrist, especially in interaction with the object. But that was kind of an interesting aspect of this, to watch him interact with the object in order to achieve targets. So in the end, he was able to achieve 7-dimensional control of the device. Here's the monkey controlling the robot fully in 7 dimensions. You can watch him attending. The real issue, we had broken equipment often, so some of the fingers would break a lot. So you may see that in some of these trials. I'm kind of cutting together multiple successful trials. One issue with this, our video was always monkey attention. It was very difficult to know when he was going to get back to it and try. It made contiguous recording very difficult. But essentially, this monkey is in real time controlling the end effector, that is the hand, just at the hand, the linear velocity and rotational velocity and grip velocity of the overall robot. And he's able to achieve targets consistently when he's trying to. So I'll let this play through here, but we kind of observed a lot of very, very interesting behaviors over, it was over a period of weeks, a few weeks usually, when this level of control will be established. And we'd watch the interaction between the robot and the objects, and the robot could hit, not him, except for him, we had a boundary region around him, but other objects in the environment. He would sometimes kind of try to do other things with the robot for no particular reason, but it was just kind of a neat thing where we could see some embodiment of the, theoretically anyway, of the robot by the monkey. All right, so after these experiments, we then looked at what the neurons were doing. So this diagram on the top left is adapted from a neuroanatomical atlas of rhesus monkeys. Essentially, and we overlaid our array locations from our different intraoperative photographs. So it's not exact, it's approximate, trying to figure out exactly where we had put these arrays in retrospect. We had 2 monkeys for this experiment. One monkey had 3 arrays, 2 actually ipsilateral to the robot, 1 contralateral, and then the second monkey just had 1 array on the ipsilateral hemisphere only. Both monkeys were able to achieve successful 7-dimensional control. Now, looking at this diagram, where these arrays are actually located, it was really the minority of the electrodes were actually implanted in the primary motor cortex. The majority were actually in the premotor cortex, both ventral and dorsal. And then the diagram on the right shows for each of these arrays, that shows each of the electrode locations, how correlated were the units picked up at each electrode with movement activity of the robot. And really it was pretty well distributed throughout this neuroanatomical space. So we didn't see a lot of correlation between, a lot of differentiation between premotor and primary motor in terms of what was being used to control the robot. We also looked at the relative contribution of individual units to linear versus rotational motion. And again, this was well distributed as well. Some individual units kind of preferred to control linear motion, some more rotational motion, but a lot of them were somewhere in between. So they were at all times encoding all or most of the parameters in this 6-dimensional, 7-dimensional space, including grip. So in any case, so kind of an interesting result and kind of says something about how the brain is able to control external devices. Okay, so this work was later translated kind of as I was re-entering medical school to a human subject study, which was published in the Lancet, Collinger and colleagues. This is also at University of Pittsburgh. So the code and concepts from our experiment were adapted pretty directly into this experiment. So this paper, my thesis is cited as a publication, and essentially they were able to quickly establish with a human, a patient who has tetraplegia. Basically, it was a very quick process. She was after the same thing the experimenters were, and was able to quickly obtain similar 7-dimensional control using the same methods, and it was a highly successful experiment. So you're watching real-time 7-dimensional control of a robotic arm using a brain-computer interface, very similar to our primate model. So this is a very good result. After this, it was also elaborated into even kind of drove that same algorithm into higher dimensions, including hand shaping. Some really interesting results kind of came from that. So it's really interesting, and you can kind of see what brain-computer interfaces could potentially do realistically for people, be it hooked up to a robotic arm like this one, or in reality, FES would be a fantastic, it'd be fantastic to have a similar FES application. So here you can see basically there's different object shapes, and the grasping is different kind of depending on the object shape. It's nice. This was later elaborated with specific hand shapes to be able to handle this kind of situation more adeptly. All right. Okay, so that's microelectrode based BCI recording mechanisms. Now I'm going to move into EEG. I have some early experience with this, but I'm going to present more recent work for BCI. So essentially EEG, as opposed to intercortical electrodes, you're reading from these large areas. These electrodes are placed on the top of the head, and you get data that looks like this. You're not looking at individual spikes. You're looking at essentially correlated behavior among many neurons. And essentially you're looking at when they are exciting together, or you can call it rhythms. It's kind of one way they describe it. For people that aren't familiar with frequency analysis, essentially you're looking at raw data from different EEG electrodes in the blue diagrams, and then below are spectrograms, or essentially the intensity of the Fourier transform, which is the intensity of activity through time at different frequencies. So essentially how much energy is there, how many neurons are firing together at certain frequencies at different times. And that becomes the basis of control used for brain-computer interface generally. So in any case, many of the more successful brain-computer interface experiments use sensory motor rhythm. The idea here is that there are a few frequency bands in which a lot of neurons excite together. Mu rhythm is a very well-known one. It's present over the sensory motor cortex, 8 to 12 hertz. They notice that there's a lot of activity in this 8 to 12 hertz spectral band that then desynchronizes, or the activity specifically in that band goes down. If you do move your arm on the other side, the contralateral side from that cortical area, if you move or imagine moving your arm or hand, that intensity in that spectral band decreases. It's more distributed throughout. So essentially, that could be used for control by virtue of being imagined movements triggering that desynchronization. The beta band is one that also has some correlation, of a similar correlation. So in any case, we're looking below. This is the homunculus along the motor cortex, the area of the brain represented by each area of the body. And then if you have an electrode over the hand area, you're imagining your hand moving or moving in reality, and that mu rhythm frequency peak goes down, or it desynchronizes. All right, so there's a group at University of Minnesota, Meng and colleagues, demonstrated an experiment that I want to show. Looks very similar to the arm movement experiment we showed, the human experiment, but fundamentally is pretty different. And so the idea here is, and we just mentioned that there's different frequency bands at which, or one in particular, the mu rhythm, where if you imagine your hand opening or closing, you can affect the activity recorded by that electrode. So imagining moving just your left hand, just your right hand, both hands, or just relaxing, no hands. That essentially creates a way to do two-dimensional control, because say moving two hands is up, no hands is down, one hand, your left hand is left, your right hand is right. So essentially that could be used to control a robotic arm in 2 dimensions. In this experiment, they had a robotic arm where they would actually, it moved in 3 dimensions, but they would actually constrain what input degrees of freedom, these 2 dimensions of brain control, would be mapped to different output dimensions in the robot. And so when a person, for instance, when the robot was moving, the first subtask was to move the robot over a target object. And essentially they would do this command structure of moving both hands, and you can see the peaks in the highlighted areas. You'll see sometimes in some of the pictures they're orange and white, and sometimes they're more blue. When they're orange and white, that's high energy or very synchronized, high intensity in that mu band. And then when it's blue, it's low intensity or desynchronized. So essentially the up target was imagining moving both hands, which is desynchronization or low activity in both those areas. No hands was high activity, shown below. Left target, essentially this is C3 is on the left hemisphere and C4 is on the right hemisphere. And so when imagining moving the left hand, you get desynchronization in C4 in the right hemisphere. And then that can be used as a left command, and then right is the opposite. So essentially that's 4 different directions, 2-dimensional control using just this. It doesn't say anything towards scale or speed, but it does provide a 2-dimensional basis for control of a robot. and it was pretty cool. So you can see here that they're doing similar reach and grasp tasks, and then actually doing a similar to the human experiment we showed previously, placement on the shelf. And this experiment was very good, but really what's being controlled here is 2 dimensions at a time. So the robot is used, the brain control is used to place their arm over the object, then control is switched to controlling up and down. And so they're just switching the planes of actuation of the robot depending on the brain input. And so this is just something to think about. When you see experiments like this, I think a lot of times in the news and other things, it's not clear of kind of what's being controlled versus what the end effector is doing. But it's important to recognize kind of that by doing things under the hood to change what is being controlled, you can actually have the same effect and produce the same output. It's kind of a version of shared control that constrains the input degrees of freedom to an output. So you can imagine kind of schemes like this may be very useful for FES control in a similar fashion. And if they're, in some research, it's not as directly stated as what I'm presenting here. They're very direct about it, of course, but like sometimes it'll be a little less direct what's actually being controlled, and what may look great on the outside may not be quite as interesting in reality. So in any case, there are more sophisticated feature extraction mechanisms. These are machine learning techniques to be able to extract more data. I mean, in what we just showed, they're able to achieve 2D control, and we could actually see what was controlling. We could see that desynchronization in certain frequency bands. But imagine kind of less apparent correlations between activity at different electrodes simultaneously, then which patterns are a little bit more difficult to pick up. So you use more advanced algorithms to figure out what those features are that could be used for control, and then sort of use those for control in real time. And there has been a 3D experiment that's been done, a proof of concept. It's kind of a little bit, it's an early experiment, and they're using a version of feature extraction to be able to do 3D control to hit cursors on a screen. It kind of reminds you of the early microelectrode recording. So, you know, it's interesting. I'm hoping, you know, it'd be great if we can develop better techniques to extract data from EEG, because it's an easier setup, it's non-invasive, to be able to control external devices. It looks like it's going to be a little trickier because of that thing I mentioned where neighboring neurons seem to prefer different directions. So the control is unlikely to be something where you're really kind of thinking about moving something in a direction. There is a sort of neuroanatomic basis for the control, the 2D control experiment I mentioned, the kind of hand on one side versus hand on the other, but then movement of that hand in a certain direction is probably not represented on the scale that EEG is able to record, at least from what we understand. But my hope is by using more advanced training mechanisms and being able to exploit different activity across the entire brain, we'll be able to produce more sophisticated control with less invasive devices. Okay, so that's it. Just want to say thanks to people involved in my research, and I really appreciate the opportunity to be here today and present to you.
Thank you so much, Dr. Clanton. Just thank you so much for explaining fairly complex content in a very clear and accessible way, and so I really appreciated that. So we do have time for a few questions, so I'll open up to people listening in. You can feel free to unmute your line and pose a question, or if you feel a bit more comfortable typing into the chat box, that's fine too. I'll watch the chat box and I can read your question to Dr. Clanton. So give you a second to think if there's any questions you'd like to ask. And maybe in the meantime, if it's okay, maybe I'll take the opportunity to ask you a question just while we're waiting.
Sure.
Yeah, so pretty basic question, but I'm just curious. So how quickly can the study participants and the human ones I'm talking about here learn to control a system with pretty good accuracy? Is it, you know, within a session, or is it multiple sessions?
I wasn't involved in that directly, but my understanding is within a day or so. It's pretty quick. I have intuitively, we had this like feeling like this was really useful, like pretty direct. I mean, it seemed to work well. It seemed, I don't know. I was not surprised when I heard the person was able to pick it up very quickly.
That's great. And I just have one other follow-up question. So my experience with similar type technologies, I work with Dr. Milos Popovich and Cesar Marquez Chin at Toronto Rehab Institute, and they do, the non-invasive approach with the EEG and FES. And my role in that work is more the interviewing the patients after 40 sessions of training and to get their perspective on the intervention and how it's beneficial or not. So more the qualitative piece. But one thing they've said to me is that there does seem to be a bit of delay. So they produce the cognitive thought, and then there's a bit of delay before the stimulation kicks in. Is that also, in your experience, the case? Is there a bit of a delay?
It all depends on the algorithm you're using. So on purpose, the microelectrode stuff we made all real time. It uses no kind of state space or memory of anything, kind of with the thought that if you turn the steering wheel left, you want it to go left. You don't want the system to double with the exception of recent cars that are doing collision avoidance. You don't really want it to check what you're doing and kind of predict based on your past input. Because that feedback and that observation, the monkey's own kind of internalization of what's going on was so important to progression in this. So I think that, I can say there was 30 millisecond delay in the, at least the command transmitted to the robot, probably a minor millisecond delay till actuation. But I felt like that real-time control was very important. EEG is a little tougher because the bin sizes for doing frequency analysis are a little bit wider. So there's going to be a little bit of delay, but depending on the algorithms used, it may be using a state space mechanism where it wants to maintain a current state or current command based on a prior kind of setup, a Markov chain or something. It's not really applicable there, but that idea. But depending on the algorithm, you may get that delay kind of introduced. And I actually feel like if any involvement I have, I want that real-time control to be there for that reason. That's really annoying when there's a delay and you're inputting commands based on something from some time ago.
Yeah, no, that's good to hear your perspective. So thank you for that. So again, line is open. If anybody would like to speak or send a chat message, feel free to go ahead.
Hi, this is Yuying Chen. Thank you, Dr. Clanton. It's really a very nice presentation. Thank you. This is not really my area of research, but I enjoy your talk very much. So I have a question is about, since this is not an area of my research, but it's really amazing. So what you can tell us what is coming soon? We can keep our eye on that in this development.
That's a good question. I think, my intuition is that this kind of algorithm could work for the microelectrode stuff kind of well for clinical devices, and I think you'll see an increase in investigational. The IRBs are very difficult, but I think based on a lot of what's been done, they're going to be easier to get. I could see an FES version of this being wildly successful if the actuation space was able to be I'm not an FES guy, I should say, but I could see that being very wildly successful, because then there might be other technical problems, but the robots themselves are kind of unwieldy, et cetera. And it might take either a portable, reliable robot to kind of break it out of the research space a little bit, or an FES application. So I think you will see more investigational studies and kind of case series kind of things, hopefully. And then for EEG, I think there's been a lot of work, and I think if they can get a package which kind of records EEG where it's important reliably, it's easy to put on. You see some commercial products with EEG, but something a little bit more, able to really do effective control like this is a little out of my area these days too, to be honest. But I could see that being more practical soon for kind of really individual experimental use, almost by kind of maker types and things like that. It's happening to some extent already. Yeah.
So you think EEG probably is more of the future?
I think it's easier. I think it's
a good direction to go.
It's great. I think there's going to be more sophisticated feature extraction algorithms, and I may be missing a whole area that I don't think I am just based on recent literature reviews. But there was probably a direction to obtain control through training. It's just going to be a little different, less intuitive than the microelectrode stuff we did. So I think neither one or the other is going to be the future. I think for the microelectrode arrays, if they're able to, get a good wireless, and people are working on this, almost like a DBS kind of setup for that. I think that's going to be the barrier for the implanted electrodes, is the craniotomy. Essentially, it's open environment.
Thanks for that question. Is there somebody else?
Good afternoon, all. This is Jean Zanka from Kessler Foundation, and I'm a Pitt grad. So, Dr. Clanton, you and I share positive feelings of Pittsburgh.
Yeah.
So I have a question about application. So you know that technology abandonment is a big challenge that we have in rehab a lot of times when there's really wonderful things that are created like this, and the challenge is to make sure that they actually get used in the ways that we want them to be used. So I imagine that in many cases the tasks that could be performed with a device like this are currently tasks that are being done with someone else's assistance. And obviously being able to do it on your own is a wonderful thing. But I'm just wondering what thoughts you and your colleagues have had about sort of the performance criteria that need to be met to make sure that this really gets adopted and used.
That's an interesting question. So there's a difference for for use of technology when people go home, because I see this all the time, clinically. There's a difference between like performing ARAT in an experimental setup versus I'm actually going to go do stuff at home with it. And a lot of the barriers to that are kind of complex. And I think if you have the right person who is motivated enough, they may just use it for the sake of using it because it's cool. To really surpass that boundary, I'm trying to think of a good example of something that kind of became practical. But things like battery life, if it's truly portable, plug and play, the plug and play qualities of the device, as well as kind of, it's usually, in the robot arms, it's always hand issues. A lot of the robot hands are less, they look like hands, but the actual movement, a lot of our movement has to do with really good thumb control, as well as, opposition with, the degree of freedom in the palm. And I think some of that has to be replicated a little bit better, more like a human hand, for that to be adopted. If we're going to really get beyond just grasping, which is fairly power grip functionally, which is that might be kind of it's a recent robot experiments I may not be as familiar with, but they're working towards these. That might be a barrier which, to some extent, in terms of really having this be a practical device for everyday common use. It's not going to work if it's like, oh, I can use this for picking things up, but then I have to have someone's help to do the next step of actually using the thing I'm trying to pick up.
Right. Well, I certainly think the observation that people can pick up the ability to use this accurately fairly quickly is really encouraging and would help facilitate its application. And using it because it's cool has value in itself. I think when people see what's possible, that's a really exciting and motivating thing. Yeah. So thank you so much to you and your colleagues for continuing to push the boundary on this, and it's really exciting to see what's happening.
Sure.
Okay, maybe time for one more question, if there's a question. So if you don't mind just waiting a second, Dr. Clanton, but also you had, I believe, shared your email address. I think if anybody had follow-up questions, is it okay if they contact you?
Yeah. Oh, yeah.
Wonderful. Thanks. We'll just stay on the line for another minute or two. If anybody has a question they want to ask with the group here, feel free to do so. Otherwise, I'll just say thank you so much, Dr. Clanton, for giving us your time and your expertise and such a fantastic talk. We appreciate it. Thank you.
Thanks a lot.
I think, Dr. Clanton, if you want to sign off, I think we can assume there's no further questions. And thanks so much.
Oh, sure thing. Thanks for having me.